checking for the audio. Let me know if the audio and my voice is very clear. We'll check the audio first. And I told you yesterday that there is some construction work going on in my building. So there is a little disturbance in between. So that's why I'm trying to check whether the audio is clear or not. Okay. I guess it's all good. And what we are going to be talking about in today's lecture are two very, very important things. That is, one is going to be photorespiration and then cell C4 cycle. Priya is here. Good morning, Priya. How are you? Is the audio clear? Just trying to check the audio first before we get started. Okay, it's fine. Okay, so what I was telling you was that in today's lecture we will be talking about photorespiration and then we will take up C4 cycle. And once we finish C4 cycle, then only we'll be able to compare the C3 and C4 cycles. Okay, so before we get started, there are a couple of things which I need to tell you, and those are the first, which is the most important, is that the subscription plan, the pricing is changing from 24th, and that this is what I was telling you from the very beginning, that in case if you want to subscribe, you need to do it as soon as possible. Rahul is here. Good morning, Rahul. Okay, audio clear, very good. Priyanka is also here. All right, let us wait for a minute. There are people joining in. Okay, so this is the schedule for our channel, that is Let's Crack NEET UG English. And we will be completing this chapter of photosynthesis very soon. There are 3 more lectures which are left, and after that, if you see the last lecture on this particular topic, we are going to have a quiz and there will be doubt solving. So for that you need to revise the lecture, come up with the doubts, and then once your doubts are cleared, then we are going to have a quiz on this particular chapter. So this is for our, and as soon as this is over, I'm going to plan for the next chapter and we will start with that. This is the other channel where I'm taking the lectures, and just 2 classes left on this channel. I'm talking about the structure of human heart and the functioning of human heart. This is Biology 101, another YouTube channel where I'm going to be talking about digestive system, complete chapter. I have already done 3-4 lectures. So after I finish this, maybe I'll start with another chapter from human physiology on this. Okay, so we are on this channel, and those who are with me, they now know me. But in case if there is some new student, I'm Dr. Neela Bakore, your biology teacher, and we are using India's largest learning platform, that is Unacademy. And this is the most important thing which I want to talk about today, is you need to install this app for few important reasons. Number one, there are students who are asking me for PDFs. So when we take a lecture, those lecture PDF is going to be available only on the app. So for that, you will have to install this app, and then you start following me. So this is how those PDFs will be available for you. Secondly, once you are on this Unacademy app, you will be able to get access to daily live classes and quizzes and all those things. not only my lectures but the lectures of other faculty members also. Now this is the screen which you were seeing for quite some time now, and I was telling you that this is going to change, and it is going to change from 24th of this month. So this is the one which you have seen earlier. See, if we are comparing this, for 1 month, after 10% discount it was 5,400. Now from 24th of October, that is 4 days from now, it is going to change. After 10% discount, that amount is going to come to 6,300. So earlier, like till 24th, it is 5,400, and after that it's going to be more. This is only for 1 month that we are talking about. If you talk about the yearly program, then also you see the difference here. Here in this case, what is available to you right now for next 3-4 days is for 1 year, 27,000 after 10% discount. And that 10% discount is available only if you are able to use this code. So if you get or use this code, then you get that 10% discount that is mentioned here. So if we are talking about the yearly program, the yearly fees earlier or till 24th is 27,000, and it is going to change to some 30,000 plus. And these are the two programs. One is called the Plus and the second is the Iconic. These are available only on the app. So you need to install the app for 2 reasons. 1, if you want the PDFs. 2, if you want to use or view the lectures of other faculty members. And 3rd, if you want to subscribe to a particular plan or program, if you want to take subscription, then you need to do it as soon as possible. And don't forget to use the code, that is Neela, so that you are able to get that 10% discount. But before 24th, if you're able to do it, there is a great benefit. Coming back to our topic, okay, there are a few messages here. Let me just read them. Study chemistry. Okay, I won't be able to help you with this because I don't know the chemistry class, about the chemistry classes. I can only tell you about my classes. Sharda is here, Sridhar is here. Okay. So those who have joined late, for them, and I am going to tell this again before we wind up today's lecture, that the subscription plan is going to change from 24th. There is a price hike. So in case if you've decided it and waiting for a day or 2, don't do that. Take the subscription as early as you can so that you are able to get the advantage of the lower pricing, which is right now. Photorespiration is the topic that we are talking about. Photorespiration, and before we start, let me just tell you what I told you yesterday also, that there is some construction work going on in the office next to mine, and that is why you might hear some disturbing sounds, like, you know, in the middle of the lecture. And if that happens, just let me know. I'm going to repeat whatever I was saying during that time. Okay. Photorespiration. Now, what exactly is meant by this? Photo is light, and respiration is a process in which oxygen is taken in and carbon dioxide is given out. If we break this word into 2 parts, then this is something which is happening in presence of light, and it is similar to respiration. So in plants, sometimes what happens is, instead of carbon dioxide, oxygen is taken in during the process of photosynthesis, and because it is similar to respiration, we call it photorespiration. When does this happen? At higher temperature, at higher concentration of oxygen. These are the two conditions when photorespiration is going to take place. And if these are the two conditions, high temperature and higher concentration of oxygen, then what happens? Rubisco, the enzyme, which normally acts as carboxylase, addition of carbon dioxide is what it is helping in. Instead of carboxylase, it starts to act like oxygenase. So if you remember, what happened in the C3 cycle was RUBP undergoes carboxylation, that is addition of carbon dioxide, and a 3-carbon compound was formed. And here the enzyme which is participating or helping is our Rubisco. This is in the normal condition. If the conditions change, in that case what will happen is RUBP, instead of undergoing carboxylation, undergoes oxidation. And here a 2-carbon compound is formed. And the enzyme which is working here is the same, that is Rubisco. Now in this first case, Rubisco is acting as carboxylase, addition of carbon dioxide, and here Rubisco is acting as oxygenase. What are those conditions when the behavior of Rubisco changes? Two conditions. Temperature is high, and oxygen concentration is also more. Now during summers, or the plants which normally grow in areas where the temperature is normally high, then in that case, instead of carbon dioxide, they are always taking oxygen. Not only this, at the end of this process, what is given out is carbon dioxide. In this case, what was given out was oxygen. So if plant takes in oxygen and gives out carbon dioxide, this is like respiration. But you have to think about one more important aspect. What is the plant taking? Oxygen. What is given out is carbon dioxide. From where is this carbon coming? This carbon is actually coming from some carbon compound which is in the plant. That means in photosynthesis, plant fixes carbon in the form of carbohydrate, whereas in photorespiration, a carbon is released. So can we say that whatever is done in the process of photosynthesis is undone in photorespiration? So photorespiration is a harmful process because the carbons which are fixed by the process of photosynthesis are released slowly. So fixed carbon is being given out, and that is why photorespiration is a harmful process. So if a plant grows in an area where the temperature is always high, so does it mean that this plant will only undergo photorespiration and photosynthesis cannot take place? Because they have to survive in those conditions, they have to show some kind of adaptations. Now, important in case of photorespiration, Rubisco is acting as oxygenase. Most of the RUBP is oxidized and very less undergoes carboxylation. So both the reactions are going to take place. This one in case of high temperature and high oxygen concentration. This is going to be the major one, and this is going to be like very small percentage, maybe 5, maybe 10, but it is so small percentage that it is not enough for the plant to survive on that carbohydrate which it has synthesized. Now, during photorespiration, oxygen is used up. This is what we have seen. Carbon dioxide is released. This is also we have seen, without production of ATP and NADPH. So there are three damages basically, or three negatives. Number one, carbon which has already been fixed by photosynthesis, that carbon is given out. So there is breakdown of some carbon compound from where this carbon is released. Second, ATP is not synthesized. Third, reducing power is also not synthesized. So if photorespiration takes place, no ATP, no reducing power is available for dark reaction to take place. So how do you think carbohydrate synthesis would take place? Okay. Now, photorespiration is also known as C2 cycle. Why is it called C2 cycle? Because the first stable compound that we get is a 2-carbon compound, which is glycolate. So you can call it glycolate cycle, we can call it C2 cycle, because so far we were talking about C3 cycle, C4 cycle we have talked about in short, and this is C2. So now we have C2 cycle, we have C3 cycle, and we have C4 cycle. Okay. So now if whether this part is clear or not, that you have to tell me. Ansab is asking, when will you start genetics? I have already started genetics on special classes, and these special classes are available only on Unacademy app, and this is what I was telling you, that you need to install this app so that you're able to take advantage of those lectures on genetics also. Ayush is asking, ma'am, is there anything called glyoxylate cycle? Glycolate is going to change into glyoxylate, but normally the name is given on the basis of the first compound. Why ATP is not synthesized? Because of ATP only light is necessary. Rahul's question. See, for ATP synthesis, there has to be a process. The electron which gets excited has to pass through a particular pathway. But here what is happening is, instead of carbon dioxide being added, like carboxylation, oxygen is getting added. So if oxygen gets added, all that in light reaction, because in light reaction also, if dark reaction is not taking place, then ADP will not be free, NADP will not be free. So if no ADP molecule, how can ATP be synthesized? No NADP molecule, so how can it get reduced into NADPH2? Ma'am, is this the last chapter? No, I gave you the schedule. There are 3 or 4 more lectures left in this particular chapter. Ma'am, in Unacademy app, you take full part of genetics in English. See, it is 80 to 90% English, but there are some Hindi sentences just to make students little more comfortable, but terminology is pure English. And yes, I will be taking the complete chapter. Okay, so I think I have full part, yes. It is going to go chapter by chapter. So right now I've started with principles of inheritance, and then we will talk about the DNA replication and one by one. All right, so is photorespiration part clear? What is happening in photorespiration? In photorespiration, instead of carbon dioxide, oxygen is taken in. The molecule which takes it is same, RUBP. So in original case, RUBP undergoes carboxylation in presence of rubisco, which is acting as carboxylase. But if conditions change, that is temperature is high and oxygen concentration is more, in that case Rubisco instead of carboxylase starts acting like oxygenase. So it is because of the behavior of Rubisco which is changing. We will talk about photooxidation a little later. This is photorespiration. Photooxidation is a biomolecule is undergoing oxidation. That is different. Here we are talking about accepting oxygen. This is conditional. Photooxidation takes place in different situation. Once we know photorespiration, we'll come to photooxidation. Okay, is this clear? Very good. So there are few people who are saying yes, clear, clear, clear. Very good. Now let us come to the pathway of photorespiration. Photorespiration involves 3 organelles. This is chloroplast, then this is peroxisome and mitochondria. So 3 organelles are involved. In the process of photorespiration. Now see what is happening here is RUBP is taking oxygen or oxygen is getting added to it, and because of which a phosphoglycolate is formed, and then this phosphate is given out, so glycolate is formed, and that is why it is called glycolate cycle, and this is a 2-carbon compound. Now this glycolate diffuses into peroxisomes. Peroxisomes are microbodies. There are 3 spherosomes, glyoxysomes and peroxisomes. These 3 organelles together form the microbodies. So in peroxisome is important because it has 2 main enzymes, that is catalase and peroxidase. So when we talk about cell structure and function, we talk about all these things. But for the time being, we need to remember that now this glycolate, it diffuses into peroxisomes. And now it changes into glyoxylate and then glycine. For formation of glycine, which is an amino acid, an amino group is required. So there is ammonia which is getting added here, it donates the amino group, and now glycine comes into mitochondria. Now when glycine enters, this is the amino acid, it enters into mitochondria and dissociates. It is dissociating into that ammonia which is recycled between mitochondria and peroxisome. And see, this carbon dioxide is given out. So what is taken in is oxygen, what is given out is carbon dioxide. So this is like respiration. And now glycine changes into serine, serine goes back, glycerate, PGA, and again RUBP regeneration takes place. The maximum times, the questions which are asked on photorespiration: number 1, which organelle are involved, conditions when photorespiration takes place, number 2, first stable compound, and 3, ammonia movement takes place where? Where is it formed? Where is it used? So it is formed or released in mitochondria and used in peroxisome. Okay? It was released here and it is used here. So this is one question which is asked. The two amino acids which are involved here, one is glycine, other is serine. So where is glycine produced? It is produced in peroxisome. Where is serine produced? It is produced in mitochondria. So we are not going to be asked this whole pathway or the complete process to be drawn. The questions which are going to be asked are going to be like parts. So the next is site of glycine production, site of serine production. These are the questions. So where is glycine produced? It is peroxisome. Where is serine produced? It is mitochondria. And now, in which organelle is carbon dioxide released? So site of CO2 release, that is again mitochondria. So what we need to remember is, which is that molecule which is going to accept oxygen? That is RUBP, ribulose biphosphate. Where is the enzyme? The enzyme is right here, and the enzyme is Rubisco. But now this Rubisco is acting as oxygenase. So in the chloroplast, Rubisco is present, and this Rubisco, instead of carboxylase, is working as oxygenase. So is the pathway clear? What is happening is oxygen is accepted by RUBP. The first compound which is formed is phosphoglycolate, from where phosphate is released and you get glycolate. And so the name of the pathway. This glycolate is a 2-carbon compound, so we call it C2 cycle. Glycolate, where is it produced? In chloroplast. Then it diffuses into peroxisomes. It enters into peroxisomes, changes into glyoxylate, and then into an amino acid, that is glycine. If it has to change into an amino acid, we need an amino group, and that comes from ammonia. Now where is this ammonia coming from? It is produced in mitochondria and it comes back into peroxisome. When is this ammonia produced? When this glycine, it diffuses into mitochondria and dissociates. It is dissociating into one amino acid, that is serine, the ammonia and carbon dioxide. And this carbon dioxide is given out as byproduct. So what was taken in was oxygen. What is given out is one carbon atom in the form of CO2. That means this carbon is coming by breakdown of some carbon compound in the plant. And from where is this carbon compound formed? That is by the process of photosynthesis. So whatever is done by photosynthesis is undone by photorespiration. Rahul has a question, ma'am, what change is taking place in Rubisco that it is acting as oxygenase at high temperature? See, what happens is, it is a protein. Rubisco is a protein, it is an enzyme, and protein's configuration or its structure is affected by temperature. The enzymes, they are normally tertiary structures, proteins, and tertiary structures have bonds, like the normal peptide bonds are there, then there can be ionic bonds, hydrogen bonds, disulfide bonds, and as the temperature increases, certain bonds, they probably break, and that is why the behavior of the molecule changes, because enzyme is going to act as oxygenase or carboxylase depending upon its active site. And if temperature is changing, the active sites are also changing. Alright, so is this part clear? The photorespiration, why is it taking place and what exactly happens when this photorespiration is taking place? Any doubt so far? All right, so Rahul, this is clear, that's good. Okay, so this is again about photorespiration, few more things which we just need to know, we have discussed them, but in case if we have to write down those points. Microbodies found near chloroplast, that is peroxisomes, are considered to be the site of photorespiration. But there are three organelles: chloroplast, peroxisomes and mitochondria. Photorespiration involves three organelles, this we have talked of. Rubisco is acting as oxygenase. Some carbohydrates are synthesized, but very less. We said majority of RUBP will bind with oxygen because Rubisco is acting as oxygenase. But some carboxylation does take place, though that percentage is very, very less. Photorespiration works to undo the act of photosynthesis. It occurs in temperate plants like rice, beans, wheat, barley, etc. These are the plants which are growing in warmer conditions. And whenever the temperature is high, these plants undergo photorespiration. So during that period, their growth is affected, because growth is directly dependent on how much carbohydrate is synthesized, how much of biomass is synthesized. So C3 plants growing in temperate region undergo photorespiration. So they have to suffer whenever the temperature is high. Now, is there any other option that the plants can totally avoid this photorespiration? So there are plants which show adaptations and they have completely avoided photorespiration, and for that they follow a different pathway which is called C4 cycle or C4 pathway. C4 cycle. It was given by Hatch and Slack, and that is why it is also known as Hatch and Slack's pathway. Named after the scientist, okay? Hatch and Slack cycle or pathway. Plants growing in warmer areas show adaptations to avoid loss of carbon dioxide by photorespiration. We were talking about this. Now, the most important thing which we need to talk about is the plants where we see these adaptations. So these adaptations are seen in maize, sugarcane, amaranthus, millets, grasses, euphorbia, sorghum, portulaca and chenopodium. This part is the most important part because there are questions asked, like they can give you options with three plants in every option and might ask you like which option has all C4 plants. So you need to remember that maize, sugarcane, sorghum, chenopodium, millets, grasses, these are the plants which have adaptations to show C4 cycle. And what will be the advantage of C4 cycle? That they will avoid the loss of carbon dioxide by photorespiration. How that happens, that we will see. And because the first stable compound formed in this pathway is a 4-carbon compound, that is why C4 cycle. So first stable compound is a 4-carbon compound, and so the cycle is called C4 cycle. So named after the scientist, we can call it Hatch-Slack cycle or Hatch-Slack pathway, and because the first stable compound is a C4 compound, we can simply call it C4 cycle. And these examples are important. What are the adaptations which are seen? There are important adaptations. One is Kranz anatomy. Bundle sheath cells are arranged in a wreath-like manner. Kranz means wreath, which means ring. I'm sure you've seen a floral arrangement where the flowers are arranged in a circular ring-like manner. That is called wreath. And when we draw bundle sheath cells, what happens is the bundle sheath cells are little thick walled and they are arranged in a ring around the vascular tissue, that is xylem and phloem. So there is a circular arrangement of these bundle sheath cells. These are bundle sheath cells and they are slightly thick walled because something special is going to take place in those bundle sheath cells, and that is known as Kranz anatomy. The second is C4 plants have dimorphic chloroplasts. We talked about it when we were talking about site of photosynthesis. Granule chloroplast and agranule chloroplast. What are granule chloroplasts? As the name tells us, grana are present. What are grana? The stacks of thylakoid. And what is agranule? Where there are only stroma lamellae, no grana present. And because we know that what is in this thylakoid or what is in the thylakoid membrane is the pigment. So if grana are present, that means pigments are also present. If no grana, pigments are not there. And why are these pigments required? The pigments are required to capture the solar energy or to absorb those photons. So if granule chloroplast is there, light reaction can take place because the thylakoid membranes have chlorophyll. And stroma anyways is there. So in granule chloroplast, light reaction, dark reaction, both will take place. Whereas in agranule, because there is no granum, no thylakoid, no pigment, light reaction will not take place and only dark reaction will take place. The stroma is anyways there. So where thylakoids are found in grana, that is granule, these are found in mesophyll cells. And agranule where grana are absent and thylakoids are present as stroma lamellae, just single membranes. And they are found in bundle sheath cells. When we talked about dimorphic chloroplast, we said the granule ones are found in mesophyll cells of C3 and C4 both, whereas agranule are found in bundle sheath cells of C4 plants. Okay. Now how this is going to be helpful, that we will see in a minute. Now we will draw this pathway. So what I'm going to do is I'm going to make two cells. This is the mesophyll cell and then we will have a bundle sheath cell. Mesophyll is the term given to all the parenchyma cells which are between the upper and the lower epidermis, and bundle sheath cells we just now talked about. These are also modified parenchyma cells and they are around the vascular tissue. So these are mesophyll cells and this is the bundle sheath cell. Now when carbon dioxide diffuses in, because here there is epidermis and epidermis has stomata and all those things, So carbon dioxide diffuses into the mesophyll cells. Carbon dioxide has to be added, accepted. But normally we directly add carbon dioxide. But what happens is carbon dioxide reacts with this water in presence of carbonic anhydrase to form carbonic acid, which dissociates into H plus and bicarbonate ions, and this is the bicarbonate which is added to some molecule. So which molecule is going to accept this bicarbonate? And that molecule is phosphoenol pyruvate. So let us write down this. This is phosphoenol pyruvate. Phosphoenol pyruvate is a 3-carbon compound, and 1 carbon is coming from this bicarbonate ion, and this results in formation of a 4-carbon compound, that is oxaloacetic acid. Oxaloacetic acid is a 4-carbon compound, and hence the name to the cycle. So the first compound which is formed is oxaloacetic acid, a 4-carbon compound. Oxaloacetic acid cannot diffuse from bundle sheath, from mesophyll to bundle sheath. So it is converted into another 4-carbon compound, which is malate or malic acid. So this malate is also a 4-carbon compound. So this 4-carbon compound oxaloacetic acid, which is formed in mesophyll cells, now comes into bundle sheath cells. So this malic acid is capable of diffusing. Now tell me one thing, addition of carbon dioxide is taking place here. Addition of carbon dioxide is carboxylation. So which enzyme is working here? The enzyme which is working here is a carboxylase, and it is called PEP carboxylase. We can call it PEPCO. So what is PEPCO? It is phosphoenol pyruvate carboxylase. So what has this PEPCO done? It has helped in carboxylation of phosphoenol pyruvate, and behavior of PEPCO doesn't change. Now where is this carbon dioxide accepted, or where is carboxylation taking place? Carboxylation is taking place in the mesophyll cells in presence of an enzyme called PEPCO. PEP carboxylase, and the compound which was formed is oxaloacetic acid, the 4-carbon compound, and now it gets converted into another 4-carbon compound, that is malic acid, and now malic acid can diffuse from mesophyll to bundle sheath. So here is malic acid. It is a 4-carbon compound. So this malic acid in bundle sheath dissociates into 2 parts. A 3-carbon compound, that is pyruvate, so pyruvate is a 3-carbon compound. Malic acid or malate is a 4-carbon compound. So 1 carbon is separated, and that is released in the form of carbon dioxide. Let us see what happens to this carbon dioxide first, and then we will see what happens to this pyruvate. Carbon dioxide is accepted by RUBP. That means carboxylation is taking place here, and phosphoglyceric acid is formed. That is our C3 cycle. And which enzyme is working here? The enzyme which is working here is Rubisco. And here this Rubisco is going to act like carboxylase. Why is it helping or working as carboxylase? You have shifted the site of Rubisco's action. It is not near the surface. It has gone deeper into the cells. As you go deeper and deeper in the cells, temperature is lower. So, in bundle sheath cells, temperature is lower because these are inner cells. One problem solved. One problem was that Rubisco's behavior changes when the temperature is high. So the cells which are on the surface, they get little heated up because of the temperature, outer temperature. But if you go deeper and deeper and deeper into the cell layers, you'll find that the temperature is little lower. So in bundle sheath, the temperature is lower. So now one problem for Rubisco has been taken care of. The second problem was oxygen concentration. So what happens in this case, mesophyll cells have granule chloroplast. And what is there in granule chloroplast? Grana. So here light reaction takes place. And oxygen is a byproduct produced during non-cyclic photophosphorylation, which is light reaction. So where is oxygen produced? Mesophyll cell. And where is Rubisco working? In bundle sheath cell. So oxygen concentration in bundle sheath is low as 1. Oxygen is released in mesophyll, and 2. Bundle sheath cell have thicker wall. So in case if some oxygen diffuses from mesophyll to bundle sheath cells, still it will not be able to enter the bundle sheath cells. And we have this carbon dioxide here, which is sort of bombarded on the head of that Rubisco. So if there is more and more of carbon dioxide, even if some oxygen diffuses in, the concentration of carbon dioxide will always be more than oxygen. Both the problems taken care of, high temperature and concentration of oxygen. And now here this same thing will happen, that is, it will change into phosphoglyceraldehyde, then RUBP will regenerate and carbohydrate will be synthesized. That means C3 cycle takes place here. So where is C3 cycle taking place? In case of C4 plants, it is taking place in bundle sheath cells. And in case of C3 plants, it takes place in mesophyll cells. This we will understand when we write down the differences. Okay, now what happens to this pyruvate? This pyruvate diffuses back into mesophyll. It is pyruvate. You have to have this phosphoenol pyruvate. So we have to change this into phosphoenol pyruvate. Pyruvate or pyruvic acid is not having a phosphate, and phosphoenol pyruvate has a phosphate. So what happens is energy or phosphate will be required. So we need ATP, and this ATP will change into AMP. That means both the phosphate bonds are going to break. See, if you say 1 ATP breaking down into 1 AMP, how many phosphate bonds are broken? 2. The third, and if we have to write it like this, this is adenosine triphosphate is changing into adenosine monophosphate. So second and third bonds are broken. 2 bonds are broken. For our calculation, can this be written as 2 ATPs change into 2 ADP? Here we have adenosine triphosphate, 2 molecules, adenosine triphosphate, and after this reaction what is happening is adenosine diphosphate. How many phosphate bonds are broken? Again 2. So for calculation purpose, can this be written as 2 ATP into 2 ADP? Because we have to calculate the number of ATPs required to synthesize carbohydrate or one glucose molecule. Okay, so if this is clear, then we will come to the ATP requirement and NADPH requirement. Okay, so is this part clear? Is C4 pathway clear? Okay, इसमें you want me to write even bigger? ठीक है, I'll try to write even bigger. But अभी जो बनाया है उसमें clear हुआ? Are you watching it on your phone? Kunal is right saying that I should be writing थोड़ा सा बड़ा लिखो. Okay, so is it like you're watching it on your phone, cell phone, and that's why probably it's a little smaller? Okay, so other slides which I'll make a little bigger diagram. Is this C4 pathway clear? What is happening in mesophyll, what is happening in bundle sheath, and why this adaptation is seen in case of C4 plants. What have they done? They have shifted, they have adaptation by which the site of C3 cycle has been shifted. C3 cycle takes place in bundle sheath cell. And for C3 cycle or dark reaction to take place, you have to have chloroplast there. Only dark reaction has to take place, so you don't need granule chloroplast. So in bundle sheath cell, only a granule chloroplasts are there, and what happens in a granule chloroplast is only C3 cycle. Okay, Rahul says to revise it in short. See, what happened is, when we talk about normal photosynthesis, those plants are called C3 plants. Two steps, light reaction and dark reaction. Light reaction we know takes place in the grana, which requires chlorophyll molecules to absorb photons. Dark reaction takes place in stroma. So light reaction takes place, ATP reducing power is synthesized, and that is used up in dark reaction. But the enzyme which is important, that is rubisco, normally it acts as carboxylase, but under certain conditions its behavior changes. There are two conditions. One, temperature is high, or if oxygen concentration is more. Then in that case, rubisco will start acting as oxygenase. So if a plant is growing in warmer areas, it will always keep going photorespiration. And photorespiration we know is a destructive process. The carbon compound which is formed is broken down, and slowly one by one that carbon is released in the form of carbon dioxide. So some plants show adaptations to avoid this problem. Avoid photorespiration. What are the adaptations? Two. One, they show Krenz anatomy. Krenz anatomy means the bundle sheath cells, they are arranged in a wreath-like manner, ring-like manner, and they are slightly thick-walled. Two, they have dimorphic chloroplast, granule and agranule. What is a granule chloroplast? It has grana and stroma. What will be in agranule chloroplast? No grana only stroma lamellae and stroma. Okay, so far clear? Okay, Kunal says yes, you're watching it on phone, and that's why probably it is slightly smaller. I'll keep this thing in mind so that next time when I draw it, it is slightly bigger. Now, in this C4 cycle, what happens is, problem is Rubisco, right? So, carbon dioxide is taken in, it enters through stomata, and it is accepted, or it reacts with water first. Here the enzyme is carbonic anhydrase. Carbonic acid is formed. Carbonic acid dissociates into H plus and bicarbonate ion. Basically, we can write it as carbon dioxide is accepted by PEP. But it is not directly carbon dioxide, it is bicarbonate ion. But that carbon dioxide is getting accepted here by PEP. And so this process here, what is taking place is known as carboxylation. And for carboxylation, an enzyme is required. The enzyme is called PEPCO, because which molecule is undergoing carboxylation? Is phosphoenol pyruvate. So it is phosphoenol pyruvate carboxylase, and we call it PEPCO. PEPCO's behavior doesn't change at higher temperature. So even if it is in the mesophyll cell, it is still going to act as carboxylase. So PEP, phosphoenol pyruvate, which is a 3-carbon compound, accepts 1 carbon in the form of bicarbonate ions, and a 4-carbon compound is formed, oxaloacetic acid, and that is why we have called this cycle as C4 cycle. Oxaloacetic acid, which is a 4-carbon compound, cannot diffuse from mesophyll to bundle sheath. So it is converted into another 4-carbon compound, which is malate or malic acid, which can diffuse. So malic acid now comes into bundle sheath cell. It is a 4-carbon compound. In bundle sheath, it dissociates into 2 molecules. One is a 3-carbon compound and 1 carbon containing, that is carbon dioxide. So in bundle sheath cells, carbon dioxide is released. In bundle sheath cells, RUBP is also present and Rubisco is also here. Now what is coming in front of this Rubisco is RUBP and carbon dioxide. So carbon dioxide concentration is more. And what about the temperature? We have shifted the site of Rubisco. Instead of keeping it closer to the surface, we have shifted it into deeper cells, that is bundle sheath cells. So temperature is lower and oxygen is a byproduct produced during light reaction, and light reaction is taking place in the mesophyll cells. So all that oxygen which is released is in the mesophyll cells. If some oxygen diffuses into the bundle sheath cells are thick walled. So there will be very, very less oxygen here. So oxygen concentration is also low. So Rubisco is going to act as carboxylase. And if RUBP undergoes carboxylation, we will get phosphoglyceric acid, then PGAL, same steps of C3 cycle. So here C3 cycle will take place. And in C3 cycle, carbohydrate is produced, RUBP will be regenerated, and Rubisco is going to act as carboxylase. This pyruvate, which is synthesized, a 3-carbon compound, has to go back for regeneration of phosphoenol pyruvate. So when pyruvic acid or pyruvate goes back, it is without phosphate. So we have to add that phosphate into it. And that comes from ATP. So ATP provides energy and phosphate. And that ATP breaks into AMP. So 2 phosphate bonds are breaking. What do? We change it to this. Instead of 1 ATP to AMP, we say 2 ATP break into 2 ADP, so that we are able to calculate it easily. If 1 carbon dioxide is taken in, 2 ATPs are required. If 6 carbon dioxides are taken in, because for glucose we would need 6 here, so 12 ATPs will be required. And C3 cycle anyways is taking place here. C3 cycle requires 12 NADPH2 and 18 ATP. So now what is the total? If you are talking about C4, this anyways is there in C3 cycle and this extra, so there will be 12 NADPH2s required and 30 ATPs. It is an expensive process, but if by spending little more energy that problem of photorespiration can be taken care of, then it is worth spending this ATPs. What is the requirement for C4 cycle? Temperature should be low, oxygen concentration should be low, and if you have to reduce oxygen, you can do one thing. Carbon dioxide should be increased so much that oxygen automatically becomes less. How is this achieved? By shifting site of C3 cycle to bundle sheath. How is this achieved? By having granule chloroplast in mesophyll and a granule in bundle sheath. And how is this carbon dioxide concentration increased? This is by this C3 cycle in bundle sheath and C4 in mesophyll. So all the problems are taken care of. Alright, so hopefully this particular part, that is photorespiration and C4 cycle, are clear. Those who have joined little late, I showed you a slide in the beginning, that is the new subscription price. I was telling you from the very beginning that this subscription price is going to go high, and it is going to be effective from 24th of October. So you still have few days where you can take advantage of that slightly lower price, and don't forget to use the code so that you are able to get this 10% discount. Okay, so Rahul, clear, very good, and thanks. Okay, so you're most welcome. I'm happy that this process is clear, because I normally see that students find the C4 cycle and photorespiration little difficult. So I'm glad that you understood it, revise it, and in the next class you can ask me doubts in case if you come across something. All right? And don't forget to take the subscription plan. You have just couple of days before the prices go up, and don't forget to use my code Neela to take the subscription. So I'll see you in the next class, and we'll continue with our same chapter. Till that time, take care. Bye bye.